who is Professor of Pharmaceutics and Drug Delivery, Associate Director of Center for Pharmaceutical Technology at University of Mississippi School of Pharmacy. He is a very important person for very many reasons. He is also founder director of a non-profit research organization, Institute for Drug Delivery and Biomedical Research, right in our country at Bangalore. In US, Dr. Murthy's research programs are well funded by NIH, FDA, and also by the pharmaceutical industries. I'm really impressed to know and also to read for the House that what a prestigious career he has, and what an important speaker we have. He has received very many awards, and so many, but he was bestowed with the Distinguished Scientist Award by American Association of Indian Pharmaceutical Scientists in just 2018. He is an endowed chair for research at Ohio Northern University, and he's also inducted as a fellow of American Association of Pharmaceutical Scientists in 2017, which is indeed a very prestigious one. Very nice. Most welcome, Dr. Narasimha Murthy, and kindly present your work to us. I'm personally looking forward to hear you and then talk what you present, please.
Sure. Thank you so much, ma'am, for a nice introduction. I'm going to share my slides first.
Your slides are clearly visible to us, and you are very nicely audible to us. Please go ahead, Dr. Murthy.
Great. I will just make some modifications here. Okay. All right. Good afternoon, everybody. Thank you so much for the opportunity to be a part of this VAIBHAV Summit. I'm going to talk about Transdermal Delivery of Veterinary Therapeutics, current status and future prospects. This is more like a review rather than presenting any hard data that we have generated, because I work in the pharmacy school and most of my work is more in the pharmaceuticals meant to treat the human diseases, not much in the veterinary diseases. But I hope and I also would like to expand my research scope and try to work more in the transdermal delivery systems meant for veterinary purposes as well. I'm a professor of pharmaceutics and drug delivery at the University of Mississippi. So the main objective of this VAIBHAV Summit is to bring together the scientific Indian diaspora across the globe in contact with the scientists in India, and they all work together for the betterment of science and technology in the country. So this we thought we should do in 2013, and then we were wondering what we should do, in what direction should we work to make this happen. So that's when we, some of my colleagues, who are like-minded, some of the senior faculty members in Bangalore, we all came together and incorporated this non-profit research organization called as Institute for Drug Delivery and Biomedical Research. I'm the founder director of this organization. So as I mentioned, this is a non-profit research organization. This is recognized by DSIR right now. It was established in 2013. We are second time recognized by DSIR as CSIRO. And then we also got the recognition by Niti Aayog, Government of India. So this is a organization located in Bangalore, mainly with the three main goals. One of the goals is to foster research in the area of drug delivery and biomedical sciences, and to address the unmet medical needs. And the second goal that we have is to encourage youngsters to do good research, good science, and to motivate the youngsters to pursue research as career, because the youngsters are moving away from science. That's the complaint I'm hearing from many senior members, members of scientific community. So we are encouraging more and more youngsters to come into this field. So we are enabling them by providing the facilities, and we are helping them to write the grants. So then establish themselves as independent researchers. That's been one of our objectives. And the third objective is, like, we do a lot of outreach activities. The outreach activities will have a positive impact on the society. So that's the goal, and that involves not only scientists, also people who are not scientific, who are serving in the public health area and such things. So just to give you an example of what we do since 2013, we enable many researchers to write research grants. We were successful in getting several grant proposals. You see that we had grants from BIRAC, we had grants from Department of Biotechnology, we had research grants from Biotechnic Research Center as well. So all these are a bunch of investigators. You see some of the investigators, they develop the product, they out-license the product to companies. Some investigators incorporated their own startup companies, and they want to pursue it further. Some ended up publishing the research, and we have had a good number of publications come out of all this research as well. So these are the activities that we perform at Institute for Drug Delivery and Biomedical Research. And right now, there is a lot of research going on in dermal transdermal delivery systems. And University of Mississippi and IDBR, the institute in Bangalore, we have an MOU, and we have a material transfer agreement in place. We work together. My main area of research is in dermal and transdermal delivery systems, so that's why most of the projects that are going on in Bangalore are also in that space right now. But we would like to expand, our areas, our area of research into other avenues as well. So coming to the main topic of the day, the transdermal delivery system. So we know that dermatological products are categorized into three different categories. Some of the dermatological products contain drugs that are intended for activity on the skin, on the surface of the skin, like antifungals and protectants and antiperspirants and such things. There are dermatologicals which are intended to penetrate into the skin tissue. The site of action is within the skin tissue. There are drugs like local anesthetics and antiviral drugs and such things administered in the form of intradermal delivery systems. What we are going to talk about today is the transdermal delivery system, in which we are using skin as a port of delivery of drugs into the systemic circulation. The target site of delivery is systemic circulation, and the target site of action could be anywhere in the body. So the transdermal delivery systems are of different types. There could be semi-solids, there could be patches, and there is a lot of research going on in the area of dermal and transdermal delivery for treating human disorders. And there's a lot of fundamental research that has been published over the last almost like half a century, I would say. So what we know from all this research is that stratum corneum, the topmost layer, the right picture shows you the skin layers. The stratum corneum is the top layer, the epidermis, the viable epidermis, is the bottom layer, and the last layer is the dermis. So the top layer, stratum corneum, and the epidermis are lacking blood vessels and lacking the neurons. So stratum corneum is really a tightly packed and tightly packed layer with very less amount of intercellular space, and all the intercellular space is occupied by lipids, and it's the biggest barrier for any drug to penetrate into the stratum corneum. So the stratum corneum allows only smaller molecules. Like when I say smaller, the molecular size is less than 500 Dalton. The partition coefficient of the drug should be somewhere between log P should be somewhere between 1 and 3, and somehow research has shown that the drugs with low melting point could permeate very well compared to the higher melting point molecules. So macromolecules pharmaceuticals is out of question. We cannot deliver passively any of the macromolecules and highly polar molecules. We cannot deliver anything like cell therapy based delivery systems and such things across the intact skin. Of course, when you compromise the skin barrier, there are a number of things can be done. So skin can be used for delivery of macromolecules and microparticles, nanoparticles, and several other things. But intact skin is highly significant barrier. So with this, so moving into the veterinary area, so as I mentioned, there's a lot of research going on in the human pharmaceutical space, but in the veterinary area, for some reason, the research has been very incremental. I would say there are some products, like for example, mirtazapine transdermal ointment is available for managing the undesired weight loss in kids, in the cats, and then there are dosage forms like twister dose type delivery system that is available, in which you can load some of the molecules that are listed on the right here: prednisolone, amitriptyline, and several other drugs also. There are patches, particularly fentanyl patches are very famous. Fentanyl patches, LHRH, and testosterone have been tested in different animal models as well for veterinary purpose. So typically, a transdermal delivery system would deliver the drug as given in this graph. The pharmacokinetic profile, the typical pharmacokinetic profile of the drug would look like this. What you see is a plasma concentration of tolbuterol in beagle dogs, at different time points. So you see the plasma concentration goes up, and then eventually it remains constant over a long period of time, fairly constant, I would say, and then it would once the patch is removed, the concentration decreases, and generally the bioavailability is proportional to the dose of the drug. So unlike in case of oral, in some of the drugs are substrate for PGP efflux transporters and such things. In such cases, the bioavailability would not be dose dependent, but in this case we generally see a dose dependent bioavailability. So like any other delivery systems, even transdermal delivery systems have ups and downs, downsides. The upside would be like creating consistent drug levels in the blood for a prolonged period of time, dose-dependent bioavailability, as I mentioned, and the pharmacokinetics is more like a IV infusion like pharmacokinetics. But the issues are, or the downsides are longer lag time. So the plasma, the steady state plasma concentration would take a long time to attain, depending on the elimination half-life of the drug. And then high variability. This is another big issue that we see in case of humans as well as in case of animals. So you can take a look at the error bars. This kind of error bars, big error bars, are pretty common in transdermal delivery plasma pharmacokinetics of drugs. So when we have so much of data in the preclinical space, for transdermal delivery systems which are meant to go into clinical phase, we generally do a lot of preclinical pharmacokinetic studies and pharmacodynamic studies, right? So we use rodent models, we use even rabbits, pigs, and dogs are very commonly used. So we have a lot of data. Can we directly extrapolate the data that we have in the preclinical element models to larger animals like cows and horses and such things? So yes, we could have done, provided if all these pharmacokinetic characteristics would proportionately vary with the body size or body weight, right? So only the volume of distribution. So the formula here is, we are calculating the rate at which the drug should be infused or it should be delivered using a transdermal patch. That's what we call as the desired flux, right? So this depends on the volume of distribution, elimination constant, and the concentration that you want to achieve, the steady state plasma concentration that you want to achieve. So generally, the literature says that the volume of distribution is proportionate to the body weight. I'm going to show you the graph in the next slide. But the elimination constant and the target plasma concentration, or minimum effective concentration, what we call as, is not really proportionate to the body weight, because of the differences in the metabolic profile of animals, species. So there's no proportionality between smaller animals to larger animals in terms of elimination constant and the MEC levels. So therefore, meeting the target concentration and then delivering the larger doses and then reducing the lag time before we achieve the CSS would be a major thing to research on, would be a major thing to address also when we want to deliver when we want to deliver the drugs via transdermal route. So not only these differences in terms of pharmacokinetics, even anatomically, if you see, there's a huge difference in terms of the thickness of the stratum corneum. Okay, there's a difference between human to sheep to cattle, right? So our pig to sheep to cattle, there's a huge difference in the thickness of the stratum corneum, and the amount of lipids. Only in case of pigs and humans, it's characterized and it is reported, and there is lack of data on the other animals. So this is some thing that one needs to work on. There is a gap in the field that can be filled in by some more research. And then there is a huge difference in the hair density. Of course, the hair density varies from 1 order of magnitude to 2 to 3 orders of magnitude in some cases, right? So in transdermal research, in case of humans, we know that the follicular pathway, what we call as follicular pathway, does not contribute significantly to the overall dermal drug absorption. But in case of animals, what could happen is if the density, the hair density increases, per se, in case of sheep, there is about 1,100 hairs per centimeter square. The number of hairs are so many that it could attenuate the epidermis and potentially increase the delivery of drugs across the skin. So the having hairs could prove to be a positive, could have a positive impact on the drug absorption in case of animals which have a very high hair density. So we have to consider these factors, before we design a delivery system, a transdermal delivery system for a specific animal. So the graph on the right shows the log correlation between the body weight and the volume of distribution. This is what I was talking in the previous slide. So not only that there are differences from one species to the other species that need to be considered, there are other things that we need to consider within the same animal, the region of application, right? So here is a picture showing the fentanyl concentration, the concentration of fentanyl in plasma with time from transdermal patches applied in two different regions in the same animal. They applied one on the foreleg region, the other in the other group they applied it on the thorax region. They found that the authors found that the drug concentration were a lot higher in case of transdermal patches applied on the foreleg region as compared to the thorax region. Okay, so there could be regional differences. This is not something that we don't know. In case of humans, we know that when you apply a patch on the back to the thigh to some other region, you would see a difference in the pharmacokinetic profile of the drug. So the same could happen even in case of animals also. So these are some of the factors. The point that I'm trying to make here is, there are challenges. These can be resolved. So there is a lot of preclinical data that is available. There is some precedence. Okay, so we can definitely explore further into development of dermal and transdermal delivery systems for the animals. So we have a lot of data in humans. Can we translate that data into animal use? So let's take a look at this slide. So in this slide, this is the percent of drug absorbed in human, and then this is the percent of drug absorbed in the pig. So there is a pretty decent correlation between human and pig. But if there's lack of data in other animals, so there's absolutely no data on the same drugs, or very few matching drugs are tested on humans as well as in the animals. So that we cannot establish a correlation like this, but only pigs and humans, we know that they correlate very well, and that's why even in the product development stage, in case if the human skin is not available, we use pig skin as a model for human skin. So during like screening the permeation enhancers or screening of formulations, we could use the pig skin to shortlist the product, and eventually once you have a smaller number of potential products, you can test them on human skin because of the availability issues of the human skin, as well as to make the product development process more cost effective. So these are some of the issues that we need to consider. There is lack of data, so there is need for generation of enough data, just the in vitro data. Okay, like looking at the drug absorption across the skin from different animal species itself would be a lot of information that might be required for development of transdermal delivery systems in the future. So again, can we translate the data from one animal to the other animal? So let's take a look at this example here. So here we have the pharmacokinetic profile of fentanyl from the transdermal patches. Basically, the same patch was applied on two different animals. So here, what you see is the dry pharmacokinetic profile of dogs. So the fentanyl in dogs. So when they applied the transdermal patch, if you take a close look at the pharmacokinetic profile, the concentration of drug picked up and then it became steady state as long as the patch was in place. So as soon as the patch was removed, the drug concentration in the plasma started declining. Right. So immediately, right after you remove the patch, the concentration of drug declined in the plasma. So this means that the drug did not form a depot in the skin. So this is the kind of pharmacokinetic profile that we observe with dogs and with the fentanyl patches. So let us see what happened when you apply the same patch in case of cats. Of course, this was like a 25 microgram per hour. They just adjusted the body weight, dose to the body weight. So here you see a pharmacokinetic profile in which, the concentration of the drug picks up and then it remains constant over a long period of time. After the patch is removed, you still see the drug concentration, significant amount of drug concentration. It's not declining. Okay, it's remaining consistent for some more, hours. So this shows that in cats, fentanyl is forming a depot in the skin. Okay, so which would be released into the systemic circulation even after you remove the transdermal patch. So these kind of differences in the nature of the molecule, these kind of differences in the skin permeability and the ability of the skin to hold on the drug in the skin as a reservoir, all these things need to be studied thoroughly before we develop transdermal delivery systems for animal use. So people have explored several other active techniques of delivery also. So far, whatever I've talked are all passive delivery, in which you just apply the formulation or a patch onto the skin, and then you let the drug permeate across the skin into systemic circulation. You're not doing anything to the formulation or to the delivery system, externally. So these are some of the examples of active delivery systems, like for example, iontophoretic delivery system. So iontophoretic delivery system is a technology in which you utilize a small amount of current. The current density is very less. You apply a very small amount of current to drive the drugs into skin. So the cartoon here is stratum corneum, viable epidermis, and dermis. So they're basically skin layers. So these are the electrodes. There is a positive electrode and a negative electrode and a power source. So you are going to match the polarity of the drug molecule, or drug ion rather, with the polarity of the electrode. So if the drug is positively charged, then you're going to put them under the positive electrode. So because of the like charges, when you apply electric current, the drug will be driven into the skin, across the different layers and into the systemic circulation. So this is a rapid way of delivering drugs, as well as you can accomplish a larger dose delivery by using these technologies. So you see a typical iontophoresis transdermal patch. These two patches, they look like a passive transdermal patch, but with an electrode at the backing. So you may have to connect them to a power source. The picture that I'm showing here is the phoracer, which is a power source, where variable power source. You can adjust the current density. Why to adjust the current density? Because the extent of drug delivery depends on the amount of current that you apply. So like in this picture. So in this picture, the authors have performed pharmacokinetic studies um, of methotrexate in rabbit model. Okay, you see that they applied three different electrical densities: 1 milliamps, 2 milliamps, and 3 milliamps. Okay, so they found that the bioavailability of drug proportionate to the amount of current they applied. On the rightmost graph, you see that there is a linear relationship between the current they applied and the AUC in milligram per liter hour. So there is definitely a lot of scope for development of iontophoresis patches. About 40 years of research has gone into this. We have a couple of patches which are intended for delivering drugs into the skin, and one or two patches which are intended to deliver the drug into the systemic circulation, approved by FDA for human use. So there is a lot of safety data available in the preclinical models. So there is a lot of scope to work on this technology. My lab did a lot of work on this technology as well. So there is another technique, electroporation, that was explored quite a bit in the last 1 century. So you see that this electroporation technique is a bit different from the iontophoresis technique. In this technique, we apply the high voltage short electrical pulses. So when I say high voltage, there's nothing to be scared of, because they're so short that they're like millisecond, microsecond length pulses. When you apply such short pulses, the stratum corneum, you see the stratum corneum.
Dr. Murthy, may I interrupt you?
Yes, sir.
I think time. No, you have to time, please.
I think I just have one more slide. I'll wind up.
Yeah, please. Thank you.
Okay, thank you. So the lipid barrier breaks down and creates aqueous pathways for the drugs to go across. So electroporation is one such technique that has been already explored for delivering vaccines in pigs. In this work, they also used microneedles, and then they compared electroporation with microneedles. And surprisingly, electroporation was found to be a lot better over any other modes of delivery of vaccine in the pig model. So the electroporation has that ability to generate the immune response, a better immune response in the animals, cutaneous immune response in the animals. So that's why electroporation could be an advantage over the other physical methods of drug delivery, in case if you would want to deliver the vaccines. Last but not the least, I'm going to talk about the polymeric microneedle technology. So the polymeric microneedle technology are basically small micron size arrays, polymeric arrays, that when applied onto the skin would penetrate only through the epidermis. It would not go deeper into the dermis. It would not really hurt the animal or hurt the subject, because it is not touching the neurons. So this is typically the micro needle array. All these the needles that you see are all like 250 to 500 micrometer diameter. They're made up of biocompatible polymers. You can incorporate the drug in them, and then when you put them into the skin, in the top picture you see that after 1 minute the microneedles are all distorted, and then within 4 minutes the microneedles dissolve and deliver all the drugs. And we use this for delivering iron in pigs to treat the anemia condition in the piglets. So we collaborated with the University of Sydney and did this work in the past. And there are other very advanced delivery systems like jet injectors, where there is no needle. You apply the formulation at using a very high hydraulic pressure that will drill the hole into the skin and deposit the drug into the skin layers. So the deeper, how deep you can deliver the drug depends on the duration and depends on the pressure, hydraulic pressure that you apply. So overall, in conclusion, I would like to say that the growth of transdermal delivery system in the veterinary field, in my opinion, has been very incremental. So there is a lot of scope for doing fundamental research as well as research in the product development field. There's a huge scope, and so this collaboration and coming together on the platform like this would definitely help bringing this science forward. And definitely the transdermal delivery system is going to be one of the major delivery systems that could be considered for delivery of therapeutics and vaccines in animals. Thank you very much. That is all I have.
Thank you so much, Dr. Murthy, for a wonderful data sharing. We shall talk at the end of the presentation, as said by Chair. Next speaker is